Okay, well, I'm supposed to speak into this cube, but I've also got notes here, so it might get a little difficult. So I'm going to start with this. Can everyone hear me here? Okay, great. So it's a pleasure to introduce Jim Ehleringer as our first keynote speaker. And it's easy to introduce Jim, not only because I was in the peripheries and a sort of perpetual lab rat at Surfer for over 10 years. But because Jim has simply accomplished so much, there's a lot to talk about. Jim did his bachelor's and master's degrees at San Diego State University, and I won't list any dates, but this is an era of Nixon and Ford and Carter. He moved on to do his PhD, and he was a predoctoral fellow at the Carnegie Institute of Washington, which my sources tell me is in Stanford, California. So he did his PhD at Stanford. He moved on to the University of Utah, where he went through in quick succession assistant, associate, and full professor, and then in 2000 distinguished professor. He's been distinguished professor since. His time there, he's supervised more than 40 postdoctoral scholars and 20 graduate students. Of course, a lot of us here will know Jim from the Isotopes series, where well over 500 students from all over the world have had the opportunity of these 2-week, very immersive isotope experiences, we'll say, where they've been able to learn from some of the best faculty and staff in the world. Jim has received too many accolades to list here, but he's a fellow of the Ecological Society of America, AGU, was elected to the National Academy in 2016. He received the Rosenblatt Prize, which is the most prestigious prize given to faculty members at the University of Utah. But moreover, he's the namesake of the Ehleringer Prize, which is, Jim was editor-in-chief at Ecology from 1989 to 2006, so 17 years. There's a prize in Ecology named in Jim's honor for the the most outstanding paper by a graduate student published in Ecology. So strive to see if you can get the Ehleringer Prize in Ecology. Okay, so Jim has accomplished an enormous amount and is a great example. So this is the Early Career Scientist Symposium, and it's easy for us to often see what people have done and have this imposter syndrome. So I just want to point out a couple of things that Jim didn't do all of this as a grad student or postdoc or even as an assistant professor. And so Jim is the director of the world-renowned Surfer Stable Isotope Lab, but the lab didn't start until 1984, at which point Jim was a full professor. Okay, and the Isotopes series, which is probably Jim's signature sort of outreach, broader impacts thing, didn't start until 1996. And so there's time to do these things. I think we can take Jim's example of just working hard, being persistent every day, setting the foundation, and then striking while the iron is hot for some of these bigger things like ISOCamp or whatever we will do. So one final thing, during the break, Dale Austin would like to get group photos of the speakers and organizers. So the first coffee break, so if we can all find Dale, he's back there in the red shirt with the camera, of course. And then with no further ado, I'll let Jim come up to the stage. You have your microphone set up.
I had a cup of coffee. Yes.
Can you hear me? Even in the back? Good. So I was asked to start off with an introduction, and I know that your eyes are much better than mine. So this is, of course, a periodic chart. And when I was an undergraduate, I was told that the mass of carbon was 12, and that chemists and physicists had all come together, and they decided that carbon weighed 12.000000000. So I turned to the TA, and I said, how come under carbon it says 12.01? The TA was stumped. Okay, so I said, oh, it must be a typo. Well, it's not a typo. The introduction here will help. Because, of course, we know that carbon has 6 protons, and we define any element on the basis of the number of protons. So 6 protons plus 6 neutrons equals 12. Okay, I can do the higher math. And we denote that as 12C, and that's stable. Okay? But what I never learned as an undergraduate was that you can have carbon with 6 protons and 7 neutrons, and 6 and 7 is 13, and that's also stable. I never learned that. But that becomes the basis of what we're doing, because what I did learn was that you could have 6 protons and 8 neutrons, and then that would be unstable, and that would be carbon-14. Well, it turns out that about 1% of all the carbon is carbon-13. And of course, 98.9 times 12 and 0.11 times 13 equals 12.011. So mystery solved. But it is amazing how often we don't talk about this in chemistry. I never learned about this in PChem. And that is our world. Our world is based on the fact that small amounts of differences in the heavy and light forms cause fractionation events. And what you're going to hear today are all these wonderful stories of how you can use these small fractionation events and learn something. Now, stable means stable. That means the composition, the amount of carbon-13 and carbon-12 on our planet hasn't changed for the last 4.5 billion years. It gets remolded and moved into different locations, but it hasn't changed. We measure that with a mass spectrometer, and you have several mass spectrometers here. Basically, we turn things into a gas, and then we use this funny notation. And the funny notation comes from the fact that in the old days, we couldn't measure absolute numbers very precisely. That's the first problem. And scientists, of which I'm a biologist, so I'll claim to be part of this, we hate small numbers. We hate small numbers. So we would measure the amount of carbon-13 to carbon-12 in a sample. And then we would reference that to a standard which allows us to be on the same international scale, an OMG. Okay? I'm ending up with a number that's pretty close to 1 with lots of decimal places after that. So what am I going to do? I'm going to subtract 1. And I still don't like small numbers, so I'm going to multiply it by 1,000 per mil. So when you see these numbers, it's not percent, it's per mil, parts per thousand. And those of us that pay taxes every year, we know about the per mil notation, but otherwise we don't use it in our society. This is probably the best application. This is a graphic that Terry Serling produced about 20 years ago, showing that there's variations in carbon isotope ratios and that the values tend to be negative. Okay? OMG, problem number 2. Biologists don't like to work in negative numbers. Get used to it. But it simply says that there's more carbon-13 in the standard material than there is in the samples. And we can see that we can separate C3 from C4 plants. There's variation in C3 and C4, and no, Bobby, it doesn't mean you can't measure the number precisely. It actually means that there's variation, and we can use that variation. So I need to finish with an example. So, of course, the example I like to use is every proud father likes to help his son through a science project. And the science project I helped my son Jeff with, Jeff is now 39 years old, so it's been a while, was we traveled around and we picked up jellies at breakfast. And we measured the carbon isotope ratios of jellies. And I told him that, I don't know if this, I told him that fruits are C3 plants, and so they should have an isotope ratio like this, and that's the distribution we saw. Oh my gosh, is right. And Jeff said, arrest these guys. Arrest these guys. It's all fake. But it turns out there's nothing wrong with that because the US doesn't have authenticity laws. Europe has authenticity laws. So these are the values that you see in Europe, and these are the values you see in the United States. So the next time you buy one of these jellies, realize that it is corn syrup that has been flavored and has a little bit of a scent. And next time you go to look for maple syrup, realize that they don't call it maple syrup. They call it syrup. Whatever syrup is. So that's the introduction. And that's to introduce you to the concept of stable isotopes and variation. And today I'm going to talk about Death Valley. And for those of you that tweet, here's the hashtag to tweet. Oh my God, hundreds of plants are killed in the desert. That ought to get some attention. Oh, this is really good for my Republican friends. Let's go conservative. You'll see what that means. But I'm going to talk about two different topics today. And I'm going to squeeze them in, hopefully, in the next half hour. So the first topic we're going to talk about is global change. Not climate change, global change. Because climate change is just a piece of the global changes that are taking place. And then I want to talk about isotopes. But I need to sort of set the biology stage first. So here's the climate setting for us. We work in this little place here called Death Valley, near Death Valley. And that's the annual temperature at our field site, which is actually not on the floor of Death Valley, over the last 45 years. 2 degrees. Not 2 degrees daytime, nighttime, 2 degrees annual change. Most of that change is caused by the lack of winter, the disappearance of winter. Now, I could plot humidity and drought and so forth, and you'd see that things have gotten drier. Things have not gotten better and wetter. So, what about other deserts? Virtually every other desert in North America sees the same thing. But if you go out into the desert, you want to ask, oh, can I see the consequences of climate change? What you're most likely going to see is not the consequences of climate change. You're going to see the consequences of vegetation change associated with invasive species. The western deserts of North America are being invaded by South African species and by Central Asian species. This is cheatgrass here shown in the top, growing into the Great Basin Desert. It forms a dense enough layer that it now connects the plants together, so when we have a dry lightning strike, we have a fire. And that fire results in the conversion from a shrubland into a grassland. And then it burns every single year. That's the scourge of the West. We have no solution for that. Of course, we're trying, but so far not very successful. That's the big change you see. Oops. Okay. This is the change you see occasionally. This is not climate change. This is the super bloom of 1978. And next week I'm going back into the desert to see the super bloom of 2019. This is where areas that don't have vegetation until we have these great El Nino events just turn absolutely beautiful for about 4 weeks. That's not climate change. That's episodes of El Nino versus La Nina. My favorite plant in the world, Celia Fern, looks like this, and they will dominate the hillside. And you will drive through there between Las Vegas and Los Angeles and say, what a boring landscape. Until this year, and you'll say, oh my God, I didn't realize there was so much beautiful vegetation there. So I did not plan this study for 40 years. But I'm going to tell you about a 40-year study because we used to have this thing called winter in Salt Lake City. And we would take spring break and we would escape winter to go down to some field sites we had in Death Valley. So what are we doing down there? Well, besides having fun and botanizing, we would set up some plots. We did set up plots, and we looked at 3 species. 2 are on the wash and 1 on the hillside. And I'll talk about the hillside species today because there just isn't enough time. They're drought deciduous, which means they lose their leaves during the drought. And they are long-lived perennials. How long-lived? We don't know. So what did we decide to do? We pulled together a team. Every team has to have an acronym. You can't go to the field. You can't go to the field and say you had fun. So our team name is Depraved. We talk about how hard it is to go to work in the field. And of course, you as geologists know just how tough it is to go to the field. But we're the Depraved team. And we go out to hillsides like this, and we simply lay out our grids, and we measure plants. I didn't realize how useful this would be when we first started. But we measure the geographic position of every plant, the size of every plant, how much leaf area they have, whether or not they're in flower, their relationships to neighbors, and we collect some leaves. Why? Because I thought eventually we might be able to do genetics on leaves. I'm still waiting for that technology to evolve, but it's coming. And then we measure protein content, and of course, what else? Stable isotopes. And as we begin to look at this, we see the same pattern that everybody who drives to the desert sees. There's incredible synchrony among the perennials. When it's a good year, everybody has a very high leaf canopy. Whether you're in the wash or the slope, it doesn't matter. There's tremendous synchrony in the deserts, both in terms of leaf cover, good years and bad years, and in terms of flowering. So this will be a good flowering year. But it's not as if it's good for one species and not good for another species. That's an important pattern to begin to recognize. And the other thing is, this doesn't tell you a whole lot about the long-term trajectories, but there are some long-term trajectories. And we only learn this from having been out there for 40 years. If you go out every year and you look at it, you say, looks the same to me, especially at 85 miles an hour as I'm driving from Las Vegas to Los Angeles because I didn't win any money and I can't afford to stop. So here we are now looking at long-term trajectories for 3 populations. We've actually measured for more, and there's one trend. The manifestation of climate change is a decrease in the number of plants. Okay, we can have a decrease in the number of plants for one of 2 reasons. We stopped having babies, and adults are dying faster. Those are the 2 options. So we begin to now look to see what's happened to these populations over time, where there's been a substantial decrease. Oh, I want to say it's not a monotonic decrease, because occasionally we do have these great burst years where there's a huge increase in the number of plants in the population. These are not large populations, probably 1,000 meters squared. So we kind of, we're going down to enjoy time away from the university. We're not trying to make a living out of this. So these are the trajectories we see. But then we begin to see some interesting patterns. Germination is not correlated well with whether it's a wet year or a dry year. What? Precipitation is not, germination is not associated with precipitation. It's associated with an El Nino year. This is an El Nino year, and we expect next week to have lots and lots of things germinating. But the precipitation, I mean, if I put my bean plants into the garden and I add water, I expect more germination associated with water. I don't see that. Mortality occurs in episodes, and mortality is not, I repeat, is not correlated with low precipitation. What? Death does not correlate with a dry year. Now, I'm not going to say that the absence of water allows plants to persist, but it's not simply correlating the amount of precipitation and rainfall. There's a lag effect here. It's not associated with El Nino effects. The correlation we find is associated with PDO, the Pacific Decadal Oscillation. Now, these are not terms that, as a biologist, I was very much aware of. But the El Nino cycle is associated with a lack of trade winds, which causes a warm water system to emerge in equatorial regions. And the PDO is very correlated, Pacific Decadal Oscillation is very correlated with El Nino, but it has more to do with temperature changes in more northerly latitudes. And they correlate a lot with each other, but there are times when they're not in the same phase. Mortality is associated with a shift from the warm phase to the cold phase of the PDO. Now, breaking this down into something a biologist can understand, which is me, what this means is that the storm trajectories are changing. It's not the amount of rain, it's the storm trajectory, which probably has to do something with the timing of that precipitation, and probably has to do something with the temperature at the time those precipitation events take place. We don't know enough yet. We don't have enough data to begin to disentangle this. But we know it's not a simple story, but we do know that the long-term trajectory is a loss of plants. And the real question is, if we have this long-term trajectory of a loss of plants and a decrease in precipitation, increase in temperature, is mortality even? What can we learn as a biologist about the mortality patterns that are taking place? And with that sort of as the background of the trajectory, now I get to introduce stable isotopes. So why would we be interested in stable isotopes? Because for the biologists in the group, you know that the fundamental problem that plants faced was when they invaded lands 400 million years ago, they went from a wet environment to a dry environment. And the first things we saw evolve in response to that wet environment were the development of a cuticle, a waxy layer to retard water loss, and eventually the evolution of something called a stomata, which is a pair of guard cells that can open and close and regulate gas exchange. CO2 and water do not diffuse through the cuticle very well, and so the role of the stomatal pore is to allow gas exchange. And so here you see a stomatal pore with two guard cells, and that regulates the amount of CO2 coming in. That fundamental change in plants can be linked to some processes. And at this stage, I like to say, I don't think of plants in terms of the Merck chart in biochemistry. I think of plants as a factory. And the factory has different employees, and they have different functions. One function of a leaf is to have a supply of CO2. So I think of economics, that's a supply function. The stomata are associated with the supply function, and the degree to which they're open depends on the degree of water stress. Okay? So if we have something which is supplying CO2, then we have to have something, a factory component within the plant that uses CO2. And that component that uses CO2 is photosynthesis. So if you have a supply function in economics and you have a demand function in economics, you have an operational point. That's the balance between supply and demand. And so the operational point here is called the intercellular CO2 concentration, and that's the CO2 which resides here, which is the balance of CO2 being sucked in for photosynthesis and supplied by the stomates. Now, I could sit there as a graduate student and measure this all day long in the gas exchange cuvette, and I'd never get my thesis completed. I probably wouldn't learn very much. But I have a good friend, and that good friend was smarter than me, far smarter than me. And he said, by the way, all the variations in intercellular CO2 are recorded by carbon isotope ratio. Oh, so this is Graham Farquhar, and this is the equation that Graham came up with about 1981. And it says, basically, that the carbon isotope ratio of the leaf depends on the value in the air, the source value, and two parameters that are constant, A and B. That's the diffusion rate of CO2 through the stomates, that's A, and the fractionation event against carbon-13 during photosynthesis, that's B, times CICA. If A and B are constant, and I know the source value of the air, then by measuring carbon isotope ratio, I have a time-integrated, flux-integrated measure of the long-term CICA ratio. That allows me to go back to this graphic here and say, oh, that variation in carbon isotope ratios in C3 plants, it's not random. What it is based on variations in the CICA ratios of plants. And ecologists have looked at these data for a while, and they've said, oh, by the way, there are these correlations between carbon isotope ratio and drought in the environment. So the values tend to be more negative in the tropics, and they tend to be more positive in dry environments. So I said, oh, good, this is going to be easy. This will be fun. Let's apply this to the stable isotopes. So we got into our hillside, and we've been collecting carbon isotope ratios on this little piece of land right here. And what do I see? Here are the data for 2017, which is a wet year, and the values for 1993, which is a dry year. Now, when I read this in the textbook, first starting to look into this, and when you would give me the carbon isotope ratio of a plant, you'd give me one number. And the number might be based on 3, 4, 5, 10 samples. One number. So all the variation I showed you in the previous graph that Turi developed is based on single value for each species, accumulating it over like 900 species. Now what I'm telling you is, wait a second. I get almost as much variation within a population as I'm seeing for all the C3 plants in the world. Wait a second. What does that mean? How can that possibly be? Well, okay, now you're looking at Jim as an artist, and that squiggly line is that C3 line right there. Okay, so it's not perfect, but it's good enough for the government. And what I want you to notice are two things. There's a lot of spread in the values that I'm measuring in this one single population. First thing is that the range is large. Okay? Second thing is, wait a second. These values, these plants are growing in the desert. And the ecological surveys that have been done say that the deserts have the most positive values, not the most negative values. So now you should start thinking, don't think ecological surveys, think biology. These are plants that leaf out only when there's rain. And otherwise, they drop their leaves. Do you think, for a second, just sort of pretend you're a leaf. Do you think for a second if water is present only for a short period of time, you're going to be, keep your stomates closed or keep your stomates open? Teleologically, I told myself, oh, if water's only around for a short period of time, you make hay while the sun shines. So it doesn't surprise me that the values are as negative as they are. Okay, they're more negative than I thought they would be. I've tested the instrument. I accept the numbers. The other thing I found was the plants seem to change from year to year. I showed you a wet year and a dry year, but what I didn't show you are the individual values. So there is a plastic component to this response. This plastic component says as you go from a wet year to a dry year, there's going to be variation in the isotope ratios. So now, in case you're bored and about to fall asleep, I need to cut to the take-home message. Then I'll come back to the rest. The take-home message is that carbon isotope ratio matters. Different values matter during something called establishment, when plants are just a couple of years old, to the performance of mature plants, and then mortality during drought. And what I'm going to tell you is that the decrease in population size that we see as the climate is changing does not result in an equal loss of all genotypes with different carbon isotope ratio values. It is directional and it is selective. So here we are looking at the populations of the plants on the slope that I'm showing you in the top right over time. We got down to about 30 plants at one time. Then we went up to almost 1,000 plants. And by the way, we never call a plant until it's survived its seedling year. Because some years we'll have 2,000, some years we'll have 10,000 seedlings. You have lots of seedlings, but they don't survive. So we have these major drought events, and the question is, what happens during these major drought events? The first thing we see is two things matter. Proximity matters. The closer you and I are to each other, the more likely we are to have roots that want to compete for the same source of water. The farther apart, there's more water space for us to use. So mortality for all plants is correlated with how close plants are to each other. Then it is correlated with carbon isotope ratio. Plants that have their stomates more closed, on average, are more likely to survive a drought event. Now, do I believe it's how wide open the stomates are? No. I believe that there probably are syndromes of characteristics that have to do with xylem cavitation and other features that are part of an adaptive suite. But carbon isotope becomes a good metric of that, and mortality is not random. So let's look to see what happened when we had that really big year and we had like 900 plants added to the population, and then we went through a couple years of drought. What happens during that drought? The first thing is that about half the plants died. And as a population of the plants that died during the first year, or during the first drought, they had a more negative carbon isotope ratio than those that survived. So that on average, statistically, plants that survived were the ones that had their stomates a little bit more closed. Oh, I forgot to tell you something, but I think it might have been implicitly obvious, that the rankings among genotypes stay constant from year to year. There's variations in the value, but the rankings stay constant. If you died in the first 5 years, I do know what your carbon isotope ratio was, and I do know what your plant size was. And what you see is a basic challenge that we have observed for many, many years in plant biology. When plants photosynthesize, they can allocate carbon to roots to get more water. They can allocate carbon to leaves for more photosynthesis. And when you allocate carbon to leaves, or leaves and stems, it has a compounding effect because you can develop more leaves and they can have more photosynthesis. But imagine it's a bit like a drug habit. I have a wide open stomata genotype and I have a relatively closed stomata genotype. The genotype that has its stomates relatively wide open has to allocate more carbon to roots during drought to feed the habit of keeping the stomates open. And so what I see is the inevitable consequence of that genetic behavior, and that is that the smallest plants, on average, are those that had their stomates more wide open. On average, statistically, those that died, but at least flowered once, had their stomates a little bit more closed. Plants that survived, as you'll see, are those that had their stomates even more closed. So these are plants that didn't make it, and we know something about their characteristic at the time. So I do note, if I go back to the last graph and look to see the plants that survived, that I find very significant correlations between the carbon isotope ratio and plant size. And it's not in the direction you would think. So you would think that the more open the stomates are, the bigger the plant because it'll allow more CO2 to come in. But realize we're going through a drought period. By allocating more to roots than to shoots, I'm not going to grow. In fact, I see something which is very counterintuitive. The largest plants are those that had their stomates the most closed. The directional selection here is so that the more closed the stomates are, on average, the larger the size. And I've tried to amplify this difference in growth by putting it on a log scale. Big difference in size. In fact, that correlation between size, negative correlation with size, and carbon isotope ratio has a value of 0.3 in 1998. And that correlation stays positive and significant through 2000, 2004, and it's gone by 2010. So there's a legacy effect. There's a significant effect of having your stomates closed that allows you to persist through these drought events and doesn't cost you anything. Appears not to cost you anything. But there's a second effect which is taking place, and to me, this is the most incredible biology effect. So the first effect I was describing is an effect that we can call it a genetic difference of stomates wide open and stomates closed. The second effect is something which is common in biology, but biologists, especially physiologists, don't study it very often. And that's called ontogenetic acclamation. That is, how has my behavior changed as a child? How do the experiences as a child mold me as an adult? So to me, the childhood is the first several years. And the event that's going to mold me is drought. So we had these great events where plants germinate, and now we don't keep life all that good. The environment changes. We go to from an El Nino to a La Nina, and now we have drought. Half the population disappears during that. How many? 30 minutes? Good, okay. So the question is, do plants respond? So here we have the isotope ratio measured in 1993, the good year, and then we come back in 1998, after a 5-year drought, and ask, how did your behavior change? And it turns out that for the most conservative genotypes, those that are on this end of the spectrum, there was no change in their behavior. The carbon isotope ratio in one good year was the same as it is in the next good year. But the more negative the carbon isotope ratio, the more the acclamation took place in those plants that survived. Not in the plants that didn't survive, because we don't know what they were doing, but those that did survive. And it's in this direction here that the more negative, the more wide open the plants had their stomates, the more they responded in a conservative way. Let's go conservative. We don't get to survive unless we change our behavior. So that suggests, as a biologist, that when I'm looking at the genetics of this system, I'm looking at two things. Was I born with a condition of open stomates or relatively closed stomates? Secondly, do I have the capacity to adjust or not adjust? Call that plasticity. Now, as a biologist, if I ask you, give me some examples of plants that are very plastic, what word would come to mind? Any word? Weedy. Weedy plants are incredibly plastic. They do well in all sorts of environmental conditions. and some plants don't have that capacity. So I think we're looking at a system which is driven by two conditions: variations in carbon isotope ratio and ability to acclimate. Because after that drought event, if I now look to say, is there any further plasticity? We never find plasticity change. They are shaped by that first drought experience. And so the carbon isotope ratio in 1998, and coming back in 2010, it stays constant. So two changes that are taking place, a climate change which is taking place, and now we want to begin to pull these together. But before I do that, you have to ask yourself, we said, is it possible that there's never any advantage to having your stomates wide open? Shouldn't there be natural selection against that? Well, to begin a better feel for this, you have to recognize that these plants are out there competing for water. I'm in your face. You want that water molecule? We're going to go fight for that water molecule. So what happens if we do the impossible beyond? We say, oh, all my neighbors, you're gone. How well do I do? So we did an experiment. Plants are competing for water. We know that we can measure growth in a very simple way, growth and reproduction. We take away the neighbors around a bunch of plants and keep neighbors for a bunch of plants. And after 2 years, notice, OMG, there's almost a doubling in the size of the plants. All I did was remove the neighbors. And there's a huge increase in the size of the plants. They started off at about 350 grams, and they're going from 350 to 369 or to 691. And the plants that got bigger had more leaves, and they had more flowers, and they had a greater reproductive output. But let's tie this to isotopes. We randomly selected these plants, so I didn't know what their isotope ratio was to begin with. And what I noticed is that in my control plants, I had lots of variation in isotope ratios and no effective growth. So you know what's coming next. For those plants that had the neighbors removed, there was a huge negative correlation between plant size and its capacity to respond if it had differences in isotope ratio. Now I see the advantage of being conservative. I survived through drought. I survived through competition. Now I see the advantage of having the stomates wide open. If my neighbor dies, I am more likely to be able to grow and fill that space and increase my reproductive output. So that would suggest that there are variations in carbon isotope ratio that can benefit plants, so that if we think of a carbon pool that comes from photosynthesis, I can allocate it to roots. If I'm stuck with this environment where I have to have my stomates relatively wide open, or I can feed it into stems and leaves for a positive gain in photosynthesis. And we can have directional selection for either of the two. So I'd like to suggest that the reason we see variation, and I'll go out on this very thin limb and say the reason we see lots of variation among lots of species is for divergent selection. For the good times, when there's lots of water and no neighbors, and for the bad times when there's competition and or drought. So we might think of variations in carbon isotope ratio, and selection should favor those plants that have a very heavy carbon isotope ratio. If there's competition, plants are competing for water, or you're in a drought, and you might expect the opposite. And if you are in an environment where the environment is not constant from year to year to year, you might expect this divergent selection. The question really becomes in the desert, what's the trajectory down the future? Unfortunately, the trajectory over the last 25 years has been drier and drier and drier. Okay, we have a super bloom this year, but it's the oddity. We are moving in an environment direction, which I think will favor the more positive carbon isotope ratios. Now, given that these plants are obligate outcrossers, it's not as if one genotype is able to persist more than the other, but we'll still have directional selection. So that's the take-home message here. We think there's divergent selection. Movement is towards plants with higher carbon isotope ratios over the last 25 years. That doesn't tell you that when I had the good period of 1910 to 1930, that I wouldn't have seen directional selection in the other direction. So I think if I was at the younger part of my career, this is what I would want to do. I've now established the background for us to go out and do the genetics. to be able to say, once I can identify the genotypic characteristics, I'm expecting that we're going to have genotypes that are predisposed to have their stomates open and predisposed to have your stomates closed. I think we're going to have juveniles which are predisposed to be plastic or not plastic. And I can walk through the scenario of why it's advantageous to be one or the other. What the plant doesn't know is what the future lies, what future lies ahead. Unless it happens to be one of these plants that's very lucky to fall by the side of the road where the water washes off the road all the time and every year is going to be a good year. That's typically not going to happen. So we have this selection here for high versus low CI ratios and this ontogenetic effect. And this ontogenetic effect is not new, but you would be surprised at how little literature there is. Most of us think that when we measure the physiology of a plant that we're growing in the greenhouse that's 6 months old, that's going to be the characteristics of an adult. And I think this and other observations have suggested you really need to understand that plants change during the developmental period. So this is the environment I expect to see next week. We are in a mild to positive El Nino year, but it has resulted in a lot more rain than you would expect for a moderate El Nino year, and the super blooms are underway. I consider every time a plant flowers to be a super bloom, but the LA Times refers that distinction to very unusual years like this. So the hillsides this year should look like that. And with that, I will stop and answer questions.
Okay, we've got plenty of time for questions.
I was trying to be on time. Catherine.
Am I supposed to be speaking into one of these?
Yes.
Is this one on? We'll see. Anyway, my question has to do with the competition among neighbors. Does that mean that these plants have any forms of allelopathy in their roots?
Good question. So allelopathy is the capacity of a plant, usually through its leaves, to release a chemical that either affects germination or affects the ability of roots to grow. So there is anecdotal evidence of allelopathy, but there is direct observational evidence. Seedlings never germinate under a mother plant. They never germinate under another plant. They only germinate in open space. So that suggests, it doesn't prove, but it is consistent with the concept of allelopathy. For Encelia, we don't have the data, but for other sister species, they do have the data that suggests that underground, when roots come together from two different plants, they don't invade the other's boundary. So there's some sort of an allelopathic signal released by the roots that says, I own this space, buddy, don't invade. Now, if the root one of plant one and root 2 of plant 1 come together, they will commingle. So that's the evidence we have for shrubs growing in the desert. We don't have those data for Encelia, but I'm working on that hypothesis. Because when we remove the data of the neighbors, all we did is remove the above ground. There's no attempt to remove any below ground material. So all we've done is to remove the plant, which would suggest that an allelopathic compound, we know this from the literature, allelopathic compounds are generated continuously. It's not like you just put it out once. And they tend to be water soluble, so they would wash away. Yes?
Are there any mycorrhizal interactions?
There are mycorrhizae. So the answer is yes, there are mycorrhizal interactions. We don't understand them fully. I was telling some people yesterday that the other half of the story here, which has not been told yet, is that there are radical changes in nitrogen isotope ratio. So either that's associated with free-living bacteria that are fixing nitrogen or mycorrhizae in some way and decomposition. We don't know. But there are radical changes. In the back?
When you talk about allelopathy and the removal of above-ground plants, suggesting
We have not done the labeled study that you're talking about. I think, Deborah, the reason I went towards allelopathy is because it's sort of prevalent in the literature since the time of Fritz Wendt. It's well known for the ambrosia species, which is a sister species to the one we're working with. So it's just the easiest solution. We have not done any critical experiment that's going to allow us to tell, except to remove neighbors. And the challenge is, of course, that when you do this experiment, you're gambling as to what the next several years will look like. And I think we have done this experiment of removing neighbors twice, 2 different decades, and saw exactly the same effect. But we have not done any of the below ground. For any of you that have been out to this desert area, someone is asking me, have I sampled the soil? No, I haven't sampled the soil. There really isn't much soil. I mean, this is gravelly down to probably 30 or 40 centimeters, and then you'll find sediments depending on the area. But it's not like you have a Michigan soil out there. And so we haven't done the critical experiments. I'm leaving that to the next generation. I mean, keep in mind this experiment has been going on for 38 years with that much in federal funding associated with it. Try to find an agency that's willing to fund things long term, and I will be applying to that agency. So we do this for fun. We have a contingency plan to keep the sampling going till year 50. So I will be coming from the nursing home on an annual basis. And we are saving all the material so that at a time when we can do the genetics, we are all set to look at genetics. And we have other data I haven't talked about with respect to carbon allocation and those sorts of patterns. But we have no direct evidence of what's going on below ground. Any other questions? Yes?
So early in your talk, you were talking about how the good years and bad years for these plants didn't correlate to the annual precipitation, right? It just made me think, is there a chance that it was correlating, though, with a pattern in the precipitation such that the precipitation was actually good those years early on when it mattered?
So the simple correlations that exist are growth and growth is better in good years. So if you take a plant and look at the relationship between growth and precipitation, those are positive correlations. The correlation with mortality has a legacy effect in that it's not this year that was bad and the plants died. It was last year was bad, and this year is bad, and next year, even if it's a good year. No, if last year was bad and this year is good, there's a greater likelihood that the plants will die in the following year. So it's as if something happens in the year before, and 2 years before the plant dies, and that sort of sets into play some sort of emotion. And usually that motion manifests itself in what we call dieback. So these are suffrutescent shrubs, and a good share of the shrub might begin to die back, 10, 20%. And then the following year, even if it's a good year, the plant is dead. And we believe in Jesus plants because we have Jesus plants. And so a plant is not dead until it's been dead for 3 years because they have been known to come back to life. Not very often, but probably 1%. So we think, 2017 was a good year. 2018 was a terrible year. No mortality. 2019 is a very good wet year. And so we're expecting to fight, despite the fact that it's going to be a wet year, we're expecting to have lots of plants that will be dead. And that death would have, I think, set into motion in the drought of 2017, 2018. So it's not, I'm trying to make simple correlations when it's actually more complicated. Yes?
So I know you said there isn't a correlation between annual precipitation, but are there shifts in the seasonality from year to year?
Oh, good question.
Yeah, because I can imagine with the ENSO and PDO cycles, that might also play a role in shifting when they're getting a lot of rain.
So good question. So the question is, are there shifts between winter and summer rain? Can I go back 800 years.
Whatever you want.
800 years ago, we had lots of summer rain in this region. With the relocation of the Pacific High, we lost summer rain. So then the other question is, are there dipole situations? And that is, does a wet summer mean the likelihood of does a wet winter mean the likelihood of a dry summer? And the answer is yes. So keep in mind that if there's summer rain coming in, the winter rains come out of the Gulf of Alaska or Pineapple Expresses off north of Hawaii. The summer rains come out of the Gulf, the Eastern Pacific. Some people say they come from the Gulf of Mexico. Most of the data shows that it comes out of the Eastern Pacific, isotopic data. In order to get that monsoon wind in, you need to have a low latent heat flux, a high sensible heat flux. So if you have a dry spring, you have a high sensible heat flux that increases the probability of the rain. So we have an El Nino year this year. We had a very dry year last year. That sort of sets into the motion, this notion that you might get more rain in the opposite season. So there is a correlation. It's not a perfect correlation. But I do believe we're sort of finishing up the analysis right now. I do believe it's statistically significant at, maybe explains half the variance. Yes?
Do you have a sense for whether there would be a similarly strong sort of isotopic ontogenetic effect in woody species or more slow-growing things?
I would absolutely expect it.
Because most of the growth chamber experiments that people especially try to use for geologic systems, they're rotating for 6 months.
Yeah. Well, I do believe, and funding cycles are to blame for this, that we failed to recognize that there are ontogenetic effects. And so we study a set of plants in a growth chamber or in a greenhouse experiment, which are short-lived. So I would not be surprised. I have tried to look at the literature of ontogenetic effects, and as always, animal biologists are far ahead of plant biologists. They've studied it more, but there's not a rich literature. Yes?
Have you measured like the storage of carbon? So if you have like a bad year and then all of a sudden a bunch of rain, they just dump all their energy to get the flower to bloom. And then because they dump all that energy, you have next year.
I mean, there could be some of that. I didn't go into the details, but most of the carbon and the nitrogen, which is stored in a plant, is actually stored in a stem. And each stem seems to operate independently from another. And so when the carbon and nitrogen content seems to get to a certain level, that triggers the flowering event. So you can have stems that flower and stems that don't flower. And so I think what happens is that there's a potential to sort of expend all the reserves and then not be prepared for the future. So to give you some idea of the magnitude, when a plant flowers, 35 to 40% of all the nitrogen in the stem disappears. I mean, reproduction is a massive event, both in terms of carbon and nitrogen. And we depend more on our analyses of nitrogen because we know that's more labile, and we know that when we're measuring TNCs in the sugars, that some of those TNCs just never are relocatable. Question in the back first.
I know this is not something that you've been working on, but it seems to me the data are just a marvelous resource for doing population dynamic modeling, and especially given the ontogenetic shifts and variability, plugging these kind of data in population dynamic models with consequences is another approach to say that's going to happen long term. Is that something you've talked to anybody about?
The first publication of this matter was the special issue last fall. That's the first time it's been published. My intention after the second paper is published to make all the data freely available to the community. There are 25,000 observations, of which we have 10 observations per plant each time. So it's a very rich data set. The next data set, which is going to be published, maybe I sort of implied this, we measure the carbon isotope ratio of every single plant. Now, the only reason I can do that is that I run a facility. And when I run out of money, we still can run samples. So we want to understand more of the carbon isotope ratio data before we publish things. But as soon as we do, my plan is to make these data fully and completely available, and hope to develop collaborations. And if not, people will have the data. I mean, I'm a firm believer that since I'm at the public trough, I have an obligation to make my data available. But in this case, really nobody paid for the data, so I don't have an obligation. Yet I do want the data to be used. The only anxiety we have is that we have 5 sites. And one of those sites, as of today, as of 10 years ago, is now within a national park. And so that means that there are some restrictions. We're grandfathered in, but there are some restrictions as to what can do. We don't do any manipulative work on these sites. We only do observations. And in a year like this, it will be a problem, because if you're traveling through the desert in a year like this, and people see 2 or 3 cars stopped, they want to stop to see why you stopped. And so people will, I mean, typically people just drive by and they don't even notice that there's a half a dozen people on the hillside. But the data will be available. Oh, and by the way, the data are in a relational database. We tend to use FileMaker. So we have very detailed metadata to go along with these observations.
Probably have time for one more question if anyone else has a question. In that case, I'll ask my question. CO2 levels. So maybe there's been, what, a 50 ppm change in atmospheric CO2 during the course of the study. So have you seen any effects of that in carbon isotope data? Or do you
So the 50 per mil change in carbon isotope ratio during this time period probably manifests itself mostly as a change in the source value, which is about 1/2 per mil. Have I corrected for that? No, not really. Can I correct for it? Yes. You might notice that I express my data now as carbon isotope ratios. I do not express my data as carbon isotope discrimination. And I do that for a very simple reason. You can make the corrections if you'd like. And the second is that most of you will not live in Graham Farquhar's world. You will not live in a world of carbon isotope discrimination. And so therefore, to have terms that are alien to different fields, particularly the animal biologists, I'm trying to not to do that. But I don't believe that the changes in carbon isotope ratio of the source have manifested themselves very much. People have done CO2 studies with Encelia at high and low CO2 down in the biosphere, and there's no reason to believe that Encelia are responding any different than any other C3 plant. Thank you very much.